Hello everyone, I hope you are having a good weekend and you are preparing well for your exams. Due to the feedback that I got on the exam dates, I have come to realize that many of you are starting with papers 1 and 2. And so I will go through this paper 2, which is the specimen paper 2 for 2022. And what the specimen paper is basically a paper that gives you an idea of what to expect in the exam. So by all means, if you are preparing for May, June, and you are starting with paper 2, I suggest you watch this video to the end. It might be a bit long, but it will totally be worth it, and it will give you an idea of the things that you might not be aware of that might come through in the exam. Have a look at the paper, and I'll admit it is relatively a bit more difficult than some of the papers that I've seen in the past 3 to 4 or 5 years. So I suggest that you watch it to the end. Don't try to be a hero. Just make sure you know the things that you do know and that you understand the context of the questions. So I'll just get right into it. Okay, here's question 1, and it is from chapter 1. So typically in paper 2, you would get these kinds of questions where you are asked to look at an image and probably identify an organelle or something. So it's very important for you to be familiar with your eukaryotic cells, your plant cells, your bacterial cells, and all of those different kinds of cells. So make sure you know the difference between all of them. So let's just have a look at this question quickly. It says figure 1.1 is an electron micrograph of part of a eukaryotic cell. So whenever you see eukaryotic cell, that means it's either a plant cell or an animal cell. And it says state how it is possible to deduce that figure 1.1 is a transmission electron micrograph and not a scanning electron micrograph. Now I'm pretty sure that this is something not many people have given a lot of thought to, that you might be asked to differentiate between the two types of electron micrographs, because typically you're often asked to distinguish between a light microscope and an electron microscope, but in this case you're being asked to differentiate between the two. So I'll just go on here and tell you, for example, that with the electron micrographs, the scanning electron micrograph allows you to see the overall detail. So for example, if you need like details about the surface of an organelle, the scanning electron micrograph allows you to do that. But if you need internal details, so like you're looking at an organelle but you want to see what's really inside of it, then the transmission electron micrograph is the one that allows you to do that. And in this case, if we look at this eukaryotic cell, please don't focus on the G because that's the labeled part. Look at the entire cell. You can see quite a lot of internal detail that is not necessarily about like separating different kinds of organelles or all of that. You can see a lot of internal detail. So that tells you already that this is an electron micrograph. So something here you can just say then is that you can't really see, you can see a lot of internal structures and like internal detail, but not like a lot of like clear surface qualities. I mean, for example, we can probably see here that this is a Golgi body, but it's not entirely clear, but we can see that it has something inside of it which has been stained dark. So that is how you can differentiate between these two. So here it says both the Golgi body and the rough endoplasmic reticulum are part of the network of membranes inside the cells. Outline structural features in figure 1.1 that identify G as the Golgi body and not the endoplasmic reticulum, the rough endoplasmic reticulum. So when you look here at G, I mean, I already said in the previous on the previous slide that G was the Golgi body. The difference between the Golgi body and the endoplasmic reticulum is that the Golgi body is not continuous. So you have these fluid-filled sacs that look something like this, but they are not connected to each other. Okay, that's what the Golgi body looks like. The endoplasmic reticulum, on the other hand, looks something like this. So there is some level of continuity on it. And the other thing with the endoplasmic reticulum is that it has a lot of, the rough endoplasmic reticulum rather, has a lot of ribosomes that are on its surface, and that is why it's rough. If it's smooth, then it doesn't have ribosomes. The third thing that you should know is that the rough endoplasmic reticulum is very often connected to the nucleus of the cell and plays a role in protein synthesis. So if you haven't watched the videos on protein synthesis, I suggest that you do so. So here the features that you can see for sure is that the sacs are flattened, so you can see flattened sacs on the image. So just flattened sacs, and they are also disconnected, all right, from each other. Excuse me, I woke up with a bit of a blocked nose. Okay, so they are discontinued. They're not connected rather. So they're disconnected. They're flattened, and something else about the Golgi body. If you look at this images I've drawn here, is that the ends tend to be a little bit swollen. So you can sort of like just write that as swellings at the ends. So swellings, and obviously the obvious one. So swellings at ends. And the obvious one here is that there are no ribosomes present. So in this case, you can see that I have written 4 different answers. Just going to tell you, there are no ribosomes on the surface, well, at least that you can see, because if there were ribosomes, you'd be seeing them like right on the surface. I've written 4 different answers, but this is a 2-mark question, so you only have to write 2 of these things in order to get full marks. So just make sure that you bear that in mind. Then let's go to the next question. It says here, calculate the actual diameter x to y of the mitochondrion labeled in figure 1.1. So I'm not going to do the actual calculation because I don't think that having copied and pasted and cropped the image that I have the actual size. But what you should do in this case, when you are given something like this and you've been told to calculate the actual diameter, in that case it's more or less saying that you need to do image length, actual length, and you know that magnification is equal to image length over actual length, right? Make sure you write that here, and you can write it out in full, like image length over actual length. Write it in full, because sometimes when you write m is equal to I over a, that might not be in the mark scheme, and so you tend to lose marks because I could be anything, A could mean anything. So make sure you write it out. You get 1 mark for writing the formula correctly. But you're being told here to calculate what's it called, to calculate the actual length. In that case here, it means that your formula would not simply stop here. You're not going to just write m is equal to I over a. You actually have to then go ahead to say if m is equal to I over a is going to be equal to I divided by m. All right. Now, how do you get I? That's the big question, right? You take a ruler. Don't go to your exam without a ruler. You take a ruler and you measure from this point here where there's a vertical line to the next vertical line, okay? And you measure that in millimeters. All right. And the question says here to give your answer to the nearest whole nanometer. So you measure this in millimeters and then you convert it to nanometers. And you know that from millimeters to nanometers is basically times 1000. All right. So that then becomes your image length. So it would be whatever you measure here times 1000 divided by the magnification. The magnification is this number here, and your answer should be, based on what I've seen, somewhere around 213. So you need to, what you need to do if you want to get full marks here is that you need to specify whatever length it is that you measured. So don't simply just say, okay, well, I've done the measurement, I don't need to tell them what the number is. So you write out the formula, which is going to be this, then you tell them image length, for example, is equal to this much. So let's say 21.3 millimeters. And if we multiply that by 1000, we are going to get 21300. I don't know why my pen is not working well here, but you get 21300, divide that by 47,000 or whatever, and then you can get your number, your actual value. So this is just to show you how that is done. I haven't done the actual calculation, but make sure that you check on that and you do it correctly. All right. Next one, many of the cell structures in figure 1.1 are surrounded by membranes. Membranes are approximately 6 nanometers to 7 nanometers wide. Describe the fluid mosaic model of the membrane structure, and there is space below for a diagram. So when they give you space below for a diagram, please by all means make sure that you draw a diagram, and not just any kind of diagram. Draw a really good diagram, because it means you get at least 1 mark for that. So describe the fluid mosaic model of the membrane. The fluid mosaic model basically means that there are there's a phospholipid bilayer in the membrane, and of course many people remember this, but what people tend to forget is that there are also proteins, transmembrane proteins they're called, that span the entirety of the membrane. Okay. So in this case, you're being told to describe the fluid mosaic structure. You're not being told to explain the features of the cell membrane. So don't explain things like there are lipoproteins, there are glycoreceptors, and so on. That's not what you're being asked to do. You simply need to say, first of all, it's a phospholipid bilayer. So after you've drawn your image, and obviously you can label your image by saying this is a transmembrane protein. And what transmembrane means is that it spans across the membrane, or in that regard, between the phospholipid bilayer. So transmembrane protein is here. So again, you can just go with the phospholipid bilayer. All right, so that's the most important one. Make sure you don't miss that. And then you also need to explain that it has these integral proteins, which are the transmembrane proteins, or you can also call them integral proteins. Now, in this case, if you look at what has been done in terms of asking you the question, they haven't really said whether or not you get a mark for drawing the diagram or not. They just said there is space below for a diagram. So it seems as though the diagram is to show that you truly know what the membrane looks like. So to be on the safe side, I would say instead of just giving 2 points here and a diagram, make sure you give 3 points so that you are assured of getting the full marks. So there are, what's it called now, the transmembrane proteins, and also you need to then speak about the fact that you have the hydrophobic tails. That's always very important when you talk about the phospholipid bilayer. Hydrophobic fatty acid tails. So make sure you mention that they're fatty acid. For some reason my pen is a bit weird today. Okay, you get what I'm trying to write. I don't know what's going on. So they are hydrophobic fatty acid tails and they are hydrophilic phosphate heads. That's all you need to know here. So again, before I just go to this last question, on that previous question, make sure that what you are describing is the structure. Again, like I said, don't mention things like the glycoproteins, the receptors, don't mention things like cholesterol and so on, because those are not part of the structure. Those are simply the features or the extra organs, if you can call them that are part of the cell membrane. So they don't make up the structure of the cell membrane. So make sure that in discussing the fluid mosaic model, you are simply discussing the key parts, which are the phospholipid bilayer, the fact that that phospholipid bilayer has hydrophobic tails and a hydrophilic head, as well as the integral proteins that are part of it, because that's what the fluid mosaic is in total. Just going to do this question now, which is the inner membrane of the mitochondrial envelope is much less permeable than the outer membrane. Suggest one way in which the structure of the inner membrane of the mitochondrion may differ from that of the outer membrane to produce a less permeable inner surface. So for something to be permeable, it means that it allows for only it selectively allows for molecules to pass through. So only certain molecules are able to pass through this membrane. That is why you would consider it permeable. And so for it to be permeable, it either has, for example, gaps. So if something's permeable, it might maybe have gaps within it that then allow for things to be able to go through those gaps, or it might have some chemical modifications that prevent certain things from going through. So if you want to produce a less permeable surface, then you would have reduced gaps between the components of the membrane, right? So reduced gaps between membrane components, that should be okay. Okay. And something else that you might want to speak about might be that you have more phospholipids with saturated fatty acids. So I think I spoke about this when I spoke about the cell membrane and transport video, where I said if you have more saturated fatty acids in the phospholipid layer, then it is more dense, and for that reason it is less permeable. So that is how you would answer this question. Okay, let's look at question 2. Question 2 is about infectious diseases and immunity combined. Typically, when you get questions on those two chapters, they are separate, but I see in the specimen paper that they've been combined. So it will be very important for you here to be able to identify how you answer the question and what you're responding to in particular, to be sure that you're using the right context. So the main cause of tuberculosis in humans is the bacterium Mycobacterium tuberculosis. Most cases of the disease involve the lungs. The bacteria can enter cells and remain inactive in a dormant state. However, the bacterium can become active to produce symptoms of the disease. In a person with active TB, the pathogen can be present in airborne droplets that are exhaled. So that, I think that's pretty obvious, especially if you realize how COVID also is transmitted. Generally, a healthy person who inhales these droplets has effective defense mechanisms in the gas exchange system to prevent infection. One example of a defense mechanism against pathogens in the gas exchange system involves the actions of macrophages. Describe the mode of action of macrophages against the bacterial cell. So macrophages are a kind of immune cell or immunity cells, or kind of white blood cells that help to destroy pathogens that invade the body by engulfing those pathogens. So you have things like the neutrophils, and if you haven't watched the videos I did on immunity, I suggest that you do so here. You have the neutrophils. The neutrophils are typically short-lived, and you also have the macrophages. So these are besides the B cells and the T cells, which have very different functions from these two. So what does the macrophage do exactly? First of all, the macrophages are able to detect when a foreign molecule enters the cell. So if you have, for example, a macrophage that looks like this, it can detect foreign molecules because it has receptors on its surface for those foreign molecules. So say, for example, you have something that looks like this, and then you get the TB cell coming in and trying to invade the body. The macrophages are able to bind to whatever they detect to be foreign and figure out what it is. So if they detect that this is a foreign molecule, they detect it through their receptors, and because of that, they are then able to engulf. So the macrophages are a kind of phagocytes. Okay. So if I were to answer this question as a student, even though it's just 3 marks, I would start by saying macrophages are a kind of phagocytes. They have cell surface receptors to detect foreign antigens. So cell surface receptors for foreign antigens. And in addition to that, once they detect an antigen, they are able to engulf. So if you say they're kind of phagocytes, that already gives you an idea that phagocytosis or phagocytosis, depending on where in the world you are, involves the engulfing of a particle. So they engulf the foreign particle. And when they engulf it, so engulfing means like they swallow it, they secrete digestive enzymes. Okay, so they secrete digestive, or if you want to be very fancy with your English here, your biology English that is, you can also say they secrete hydrolytic enzymes. They secrete hydrolytic enzymes that destroys the foreign pathogen. Okay, if you're able to say all of that, you only need 3 points here, but if you're able to say all of these, you should get 4 points. You should get 3 points rather, completely, and that should be fine. Sometimes tuberculosis survives within a macrophage instead of being destroyed by the macrophage. So in this case, how would it say suggest one way in which tuberculosis may survive within a macrophage? Well, sometimes you do have situations just like when you have antibiotic resistance in microorganisms. You can also have certain antigens within the cell that are able to secrete inhibitors. So even though the cell engulfs them, they secrete inhibitors that go outside of them to protect them from the digestive enzyme that the macrophage might secrete. So in this case, I would say, for example, it means that it can produce inhibitors that deactivates the hydrolytic enzymes, or sometimes they can also form what we call a resistance spore. So if you so I'm just going to write here, secrete inhibitors, can secrete inhibitors. And remember, the inhibitors are against the hydrolytic enzymes, so make sure to write that out. Or it can form a resistant spore. And I'll just explain to you very quickly what a spore might be. using something that you might not expect, so a resistant spore. So when we discussed, I think it was transporting plants, we spoke about how no, probably not transporting plants. I think this was actually closer to chapter 18 or 19. But we spoke about how when you have a seed of a plant, the seed is in a dormant state, right? So if, for example, you're giving seeds for tomatoes, those seeds stay that way until you put them into soil and you nourish them with some water, and so they start to take in water, and once they take in water, they start to break down whatever starch or carbohydrate they have within them and start to germinate to form the plant. A resistant spore is something like that. It is basically a protective shell that is formed around the organism so that whatever is happening outside of the organism does not affect it until the organism finds a more suitable, warm, welcoming environment in order for it to germinate or to reproduce itself. So pathogens are also able to do that, and that is one of the ways that M. tuberculosis might be able to survive within a macrophage. It goes on to ask us that a healthy person has other defense mechanisms in the gas exchange system to prevent bacteria from entering the cells. Describe these defense mechanisms and explain how bacteria in inhaled air are prevented from entering the gas exchange system. Now, this is something that you learn, and this is now making me realize that this question also is incorporating chapter 9. So this is a combination of chapters 9, 10, and 11. So first things first, you need to think of the fact that in the gas exchange system, you have, you can think of your nose. I mean, you can actually start from the nose, that your nose has hairs that prevent certain particles from going in, but those hairs typically prevent things like dust and, like, you know, solid particles and so on. So maybe that might not be the best answer here, but one of the defense mechanisms that's very sure is mucus. So think about the fact that if you walk into a foreign room, into a room rather, that might be dusty or something, you start to sneeze, or your nose becomes really watery and you start to produce mucus, and you need to blow your nose. That mucus is actually a defense mechanism, and it is secreted by the goblet cells in the so the goblet cells secrete mucus. So here you only need to write 3 of these, okay? So goblet cells secrete mucus. That's one defense mechanism that you can refer to. You can also speak of the fact that the mucus traps the foreign organisms. And I'm going to explain to you very quickly, foreign bacteria, why you can't use the hairs in your nose here, because they are very clear that it's about bacteria, and most of the time the hairs in your nose are unable to trap bacteria. What they trap really well are things like dust particles and very fine particles that may be in the atmosphere, as well as things like pollens. So those are the things they are typically able to trap. So that is why you would go to this detail of like mucus, and then the mucus traps the bacteria, and once the mucus traps the bacteria, you have what you call the ciliated cells. And I'm pretty sure if you've studied gas exchange and smoking, then this should be easy for you to follow. They have ciliated cells that then, you know, clear up the mucus to move it away from the lungs so that the lungs don't get clogged. So those are the defense mechanisms. I also want you to take note of how this has been asked here. It says describe these defense mechanisms and explain how bacteria are prevented. So it means that when you're structuring your answer, you're structuring it with the view of bacteria and not just any general organism, which is another reason why you wouldn't refer to the hairs in your nose as your typical answer here, because again, that doesn't necessarily trap bacteria. So you only need to write 3 things here and you will get 4 marks. Oh, let me erase this here so we can see that one. It says in people with a weakened immune system, M. tuberculosis can infect other organs and tissues such as the kidneys and joints. Suggest how the bacteria may spread from the lungs to the organs and tissues. This is very easy. It can spread through the blood, so that's one way to do it. And of course, if it forms things like a dominant spore, like a dormant spore system, then it can also spread through the macrophages and escape at different points. But I'll say the very clear intuitive answer here would be for you to just say it can spread through the blood. and that would be fine. You don't need to. So again, something about the biology exam that I often tell students: don't be so tied to the idea of sounding smart so that you can impress the marker. In as simple words as possible, present your answer, make sure they're correct, and that should get you the marks. Okay, don't think too deeply, too far, trying to impress. Just think about trying to be correct. So next one says TB in humans can be caused by another species of bacterium, which is M. bovis. States the mode of transmission of this pathogen to humans. So if you've watched the videos on infectious disease, you will know that M. bovis is the kind of tuberculosis species, right? Yeah, the bacteria species that is found in cows. So bovis refers to cows. So you might be able to get M. bovis from eating things like contaminated meat or drinking contaminated milk, or milk that has not been treated. So when we treat milk, we call it pasteurized milk. And pasteurization basically means that we put the milk through high pressure and temperature in order to kill whatever organisms might be in it. So one mode of transmission might then be unpasteurized milk or eating contaminated meat, considering that this particular strain of mycobacterium is typically found in cows. Okay, let's look at this next question over here. So here it's talking about an antibiotic. So the antibiotic is rifampicin, and I think in the video I did on immunity, I mentioned I went a little bit over this particular antibiotic. So if you haven't watched those videos, again, I suggest that you do. It is one antibiotic that is used to kill mycobacterial cells, and although it is effective, it is the first antibiotic to which resistance develops. The way it works is that it binds to a section of RNA polymerase that is attached to the DNA template strand. And then it says, explain how binding to RNA polymerase allows rifampicin to kill mycobacterial cells. The way to think about this question is, first of all, rifampicin is binding to RNA polymerase. What does RNA polymerase do? It makes mRNA from the DNA. Okay, if you make mRNA, what is the point you're trying to achieve? You're trying to make proteins. Okay, and if you're able to make enough proteins and assemble them, you will make new cells. All right, so in this case, let's think of this here. It says it binds to a section of RNA polymerase, and how does that then allow it to kill rifampicin? So first of all, it ensures that no mRNA is made. Okay, so it prevents the synthesis of mRNA. That's one answer. The second thing that you would look at here is that, because you have to give 2 points, okay? So it prevents the synthesis of mRNA, and if it prevents the synthesis of mRNA, it prevents the formation of proteins, okay? So it prevents protein synthesis. That's more or less the big thing. And if it doesn't make any proteins, it means that the bacteria cannot make new cells. And besides not being able to make new cells, it also means that the bacteria cannot make the cells that are necessary for it to grow as well. Because remember, just copying the DNA doesn't necessarily mean we're trying to make new cells. It might also be about making cells that are required for growth, for synthesis of certain compounds, and so on. And so rifampicin prevents these two key things. So again, the way to think about this question is to ask, why is rifampicin binding to RNA polymerase? What is the role of RNA polymerase in general? And that will work you to the answer. The question continues, which, I mean, this is very interesting because, again, typically you don't get questions that are this long or this detailed for chapter 9 and 10, but because they've combined it, it is a very long question. The standard treatment for TB continues for 6 months. Initially, 4 antibiotics are prescribed. It is then reduced to 2 of the 4 antibiotics, which is rifampicin and isoniazid, if the person responds to treatment. A person with multi-drug resistant TB does not respond to treatment with rifampicin and isoniazid. The treatment for multi-drug resistant TB involves other antibiotics and can last for up to 30 months. This shows the number of reported cases of TB and drug-resistant TB in the Southeast Asia region between 2005 and 2014, as published by the World Health Organization. So obviously you read that, but if you're looking at it, you don't know what exactly you're looking at. So what does the question ask us to do? It shows that there is an overall increase in the total number of reported cases for both TB and MDR-TB. So this is MDR-TB over here, and this is TB. And you can see for TB it goes up to the millions, and for MDR-TB it's in the tens of thousands. So it says here, describe the differences between the trend in the total number of reported cases for TB and the trend in total number of reported cases in MDR-TB as shown in table 2.1. So in this case, you're not required to do any hectic analysis. You simply need to pay attention to the figures because it says describe the differences between the trend. So it means you're looking at 1 year to the next, what exactly is happening. So if we look at TB, let's start with TB. I'm just going to erase some of these, and I think I'll switch to a blue pen for once. If we look at this, so in 2005 we had, let's just say, 1.9 million cases, and then we had 2.1 million cases, 2.2, and again this is maybe approximately 2.3. This stays at 2.3, this is 2.3, this is maybe 2.4 if we approximate, 2.3 again, 2.3 again if we approximate, and then this is around 2.5. So the difference here is not like there isn't a large jump in the number of cases that you're observing. But if you look at MDR-TB, you start with 68, and then you go up to 779, and then it's 918, 1,717. And you can see that the figures at this point between 2011 and 2012, the figures more than double. So the difference in trend here is that you first of all need to say the rate of increase of MDR-TB. So the rate at which MDR-TB occurs in the population increases at a higher rate than the TB cases. So rate of increase is higher for MDR-TB than it is for normal TB. Something else that you also want to say is that you want to look at the places where there was a difference in things that just, that seemed different. So if you look at TB, we've had an increase in trend the whole time until we get somewhere down here. I think so here it's 2,358, then it decreases to 2,331,000, not 2,000, and then it decreases again. So you can speak about the fact that there was a decrease from between 2011 and 2013 in the occurrence of normal TB, but there was an increase rather in the same time period for multi-drug resistant TB, and that should give you full 2 marks here. So again, remember rate of increase for normal TB. So rate of increase for normal TB is less than multi-drug resistant TB, and also decrease between 2011 and 2012 for normal TB. Decrease in TB between 2011 and 2012, so that would give you 2 marks. Many social and economic factors. These are the kinds of questions that you get that you typically used to get, but as you can see, it has become a little bit more complicated. Many social and economic factors need to be considered in the prevention and control of MDR-TB. Outline 3 of these factors. So again, MDR-TB is speaking about the fact that like these bacteria are beginning to develop resistance to the antibiotics that are typically used, and so something else that you need to mention is that you need to educate people. One of the reasons why we have antibiotic resistance is people are not well educated on the fact that if they don't finish a typical, like a complete bacterial course, a complete antibiotic course, then that might lead them to causing the bacterium to develop resistance. So educating people about completing their antibiotics, that would definitely be a good thing. Also minimizing the use of antibiotics in terms of how you prescribe antibiotics, because sometimes people use antibiotics even for things that don't require antibiotics, and so for that reason you might have an issue there. And I think one of the other costs that I would suggest is that you might need to speak about maybe thinking of developing new drugs that will help with these, because antibiotics have become resistant. So you might want to maybe think about natural medication from plants and all of those things. With these questions, these kinds of questions, you can typically go a bit out there. You don't have to be too biological. You just need to think in the context of someone operating in a population where there is drug resistance. So that might help. Okay. Let's look at question 3. So we've only done 2 questions and we've spent 34 minutes. So I try to keep these videos short, but I also know that you require the detailed explanation so that the questions make sense to you. So I don't mind going a bit longer, but I hope that you do watch the videos and you are following. So question 3, it says here, the unicellular fungus, I'm not going to say that, is found in dairy products. It is a safe microorganism to culture for the extraction of the enzyme lactase. Lactase catalyzes the breakdown of lactose, which is a sugar that's found in milk, and the reaction catalyzed by lactase is shown in figure 3.1. So this is lactose over here, and you can see that lactose is made of 2 monosaccharide sugars, and it says there plus Y. And then lactase comes, and you can see then that the reaction produces galactose and a product Z. It says here then, describe the reaction that is characterized by lactase. Use figure 3.1 to help you in your answer. Identify Y and product Z. So you have 4 marks to do that. I think I'll go back to my red pen. This blue one is a bit not as bright as I'd like. What this question requires you to be able to do is to be able to identify alpha glucose. Okay, so alpha and beta glucose. I'm just going to write alpha plus beta glucose. Those are important. Also be able to recognize galactose. Okay, and for these kinds of questions, typically you should know fructose. But I'll tell you now for free that fructose is not here. Okay, so lactose, something else that you might also want to know is, what is lactose made of? Lactose is made up of galactose and glucose. All right, so I mean, just knowing that, then you should be fine to answer this question. I'm pretty sure if you're watching this video by now, you're able to answer the question. So first of all, describe the reaction that is catalyzed. Whatever reaction is catalyzed here, because it involves the breaking of the glycosidic bond, which is this one, is a hydrolysis reaction. So you can say this is a hydrolytic or hydrolysis reaction. Okay, so this is hydrolysis. Y, if it's hydrolysis, it means that you're adding water. So Y is equal to water. Okay, so it's a hydrolysis reaction, and what I would say here is hydrolysis involves, this is a hydrolysis reaction, it involves the breaking of a bond, of the glycosidic bond, because you have to be specific in this case. You've been given the figure because it says use figure 3 to help you. So this is a hydrolysis reaction, breaking of the glycosidic bond, break glycosidic bond. All right, so if it's breaking the glycosidic bond, then you can say Y involves the addition of water, and if Y involves the addition of water, then you produce galactose, and what is product Z? Product Z in this case is going to be alpha glucose. And how do I know that this is alpha glucose? I'm going to tell you now. I know this is alpha glucose because the number 1 carbon, which is the carbon over here, has its hydroxyl group at the bottom. That's the way I recognize alpha glucose. Okay, if you want to recognize fructose, fructose has a bit of a house-like structure that's a little bit different from the way glucose looks. Glucose has a flat top, and so does galactose. So this is alpha glucose. So it is a hydrolysis reaction that involves the breaking of the glycosidic bond. Y is water, which helps with the breaking of the bond to produce galactose, and alpha glucose, which is product Z. That should get you 4 marks. Okay, so I see here they have also combined enzymes with this question, with biological molecules. On a commercial scale, immobilized lactase can be used to produce lactose-free milk. One of the products of the reaction shown in figure 3.1 acts as an inhibitor of lactase. This is an example of product inhibition. Explain why product inhibition is useful in K-lactase when lactase is acting as an intracellular enzyme, but is a disadvantage when extracted lactase is used free in solution for the production of lactose-free milk. So I'm just going to tell you very quickly that product inhibition basically means when the enzyme reacts on a on a substrate to produce a product, the product increases in amount until it gets to a certain point where the product itself binds to the enzyme to inhibit it. So it's a way of the product telling the enzyme that you have made enough product, so you need to stop functioning. This is an advantage in intracellular function because it helps to prevent the unnecessary buildup of certain products and allows for balance of the product within the cell. The disadvantage when you use it free in solution for producing lactose-free milk is that the enzyme stops working. So it means that you lose productivity of the enzyme in the process because the product starts to bind to the enzyme. So that, just writing that will give you 2 marks. The second thing here, it says suggest how using immobilized lactase for the production of lactose-free milk helps to reduce the problem of product inhibition. So when you immobilize an enzyme, you're basically putting it within a shell to protect it that way. So one of the ways that it helps to reduce the problem of product inhibition is that the product is less likely to bind to the enzyme in order to inhibit it because the enzyme is contained within a protective shell. The first large-scale production of lactose-free milk with an immobilized enzyme used lactase trapped in cellulose triacetate fibers suggests one feature of cellulose triacetate that makes it useful as an immobilizing material. So if you want to immobilize an enzyme within a material, the most important thing is that that material must be on-reactive. Okay, so in this case you only have to give one answer. So I would say just go for the fact that it is unreactive and so it doesn't interfere with the actions of the enzyme. Another thing that you also want is that you want your immobilizing material to be insoluble in whatever solution it is that you're going to put it in. So if it is soluble, then it basically means you put your enzyme within the shell. When you throw it into the solution, the shell dissolves and then the enzyme is free. So there was no point in you doing that immobilization. But if it's an insoluble material, the enzyme stays within the material because the material is insoluble, so it acts like a good cage. Then the last question here is that when developing an enzyme catalyzed reaction for use in industry, the progress of the reaction is studied. Outline how the progress of an enzyme catalyzed reaction can be investigated experimentally. This again is just so very different from what you typically get in paper 2, because paper 2 is one of the easiest papers, but I see it seems as though in 2022 you are going to sweat a little bit. And I would tell you also that the specimen papers are typically easier than what you would get in the exam. So please put your nose to the grind and make sure you study hard. So if you want to investigate an enzyme catalyzed reaction experimentally, first of all, think of the factors that affect enzyme activity. So you want to think of things like the temperature. Okay, so you need to keep the temperature constant. You need to keep your pH constant as well. All right. Obviously, you need to take samples of your reaction so that you can figure out the product. So take samples at interval. That's a very important one. Ensure you know the substrate concentration so that you can know how much you have broken down, and obviously you also want to measure the product, measure amount of product formed. In this case, because you haven't been asked to draw a graph or anything, but typically you can then draw a graph that shows maybe either enzyme concentration or something, or product formation. You can show product concentration and then a rate of reaction. Okay, I don't know why my pen does that, but I hope you get the gist here. Now let's go to students' favorite subject, which is transporting plants. It says figure 1.1 is a diagram of a section through part of a young root. Please, when you are studying chapter 7, do not ignore the diagrams, okay? Chapter 7 is actually one place where you need to look at the diagrams. Besides chapter 1, you need to look at the diagrams in chapter 7 and make sure you know them. Know what the root looks like, know what the shoot looks like, know how to do your biological drawings. Those are all very important. Okay. Describe the pathways by which water passes from the soil to the cells of the cortex shown in figure 4.1. So this is the film of water. This is the root hair. Okay, this is called a root hair over here. So how does water get through? Well, first of all, you need to know that there are two pathways in the way water travels through plants, and that is the simplest pathway or the apoplast pathway. Okay, so either the simplest pathway or the apoplast pathway. So you need to mention that water can travel either through simplest or through the apoplast. Then you also need to mention here that water will enter into the root hair. This is very important. The water enters into the root hair and it travels so it gets into the root hair through osmosis. That's very important as well. So water enters the root hair through osmosis and it travels all the way until it reaches the cell over here, and then it will travel through the cytoplasm or between the cell walls or between the plasmodesmata matter. So I'll say that again because I realized I've just rambled through it. So first of all, you start by saying water can travel either through the simplest or the apoplast pathway. Water will enter the root hairs. Okay, so enter root hairs. Once water enters the root hairs through osmosis, so you need to make sure you also mention that, through osmosis. And please write your words out in full. Don't write through as THRU. Water enters the root hair through osmosis, and then it travels through the cytoplasm of the root hair. So it travels through cytoplasm. I'm about to write through the exact way I told you not to write it. Travels through the cytoplasm, and then when it gets here to the cell over here, this group of cells over here, it will either travel through the plasmodesmata. Okay, it will either travel through the plasmodesmata or it goes through the cytoplasm of the cells, but it hardly goes through the cytoplasm of the cells. Sorry, that's wrong, because if it does that, it's going to interfere, and there might be losses and so on. Sometimes it does that, but in this case, you can say it travels through the plasmodesmata until it gets to the cortex. So when it gets to the cortex, then it can travel through a different means, but traveling through the plasmodesmata is most likely your correct way of putting this, and that should give you 4 marks. All right. Okay, figure 4.1 shows the location where mineral ions in the soil enter the plant. There is a greater density of mitochondria in cell X than in the cell of the root cortex. With reference to the uptake and transport of mineral ions suggests why there is a greater density of mitochondria in cell X than in a cell of the root context. So it says with reference to the uptake and transport of mineral ions. So over here, that's the soil particle, and you have mineral ions, and then there is cell X. First things first, I think this is really easy and straightforward. If you're being asked why there's a lot of mitochondria somewhere, then it means that some form of energy is being used there. So that already gives you some context, right? Even if you don't know anything about the uptake of mineral ions, just the fact that you have a lot of mitochondria means there's a lot of ATP that's being produced. But in this case, because they have said make a reference specifically to the uptake of mineral ions, you don't need to say, oh, it's used to produce energy, because that doesn't tell you anything. You are being asked to explain how that helps with the transport. So in this case, you then say mitochondria is required to produce ATP for active transport. Okay, so active transport is the first thing that should come to your mind, and then the second thing that you need to talk about is that the mineral ions tend to travel against the concentration gradient, and so for this reason you need the mitochondria to provide energy. All right, so mitochondria provides energy, and that should give you 2 marks there. Then the sinoatrial node and the atrioventricular node are 2 regions of the heart. Explain the role of the SAN and the AVN in the cardiac cycle. So first things that you need to know with the SAN is the pacemaker. So whenever they say someone's got a heart condition and they've had a pacemaker inserted, what the SAN does is that it basically regulates the heartbeat. All right, and the way it does that is that it will initiate the excitation wave. So it sends out the excitation wave to where it should go so that you can have the heartbeat taking place. So it initiates the heartbeat as a pacemaker. So that is the SAN. The AVN, or the atrioventricular node, on the other hand, relays impulses to the ventricles, and it also helps to conduct the excitation wave through the heart. So in this case, because it's just 3 marks, I would say you can distribute the points based on what you know. If you know only one thing about the AVN, then write one thing and then write 2 things about SAN, or you can move it around whichever way that you like. But make sure that you write about both. Don't write only about one and write 5 points about that one, expecting that that would get you 4 marks. You won't get 4 marks for that, all right? You will get marks for writing. So if, for example, it's 3 marks and you've written 3 or 4 things about only the SAN, you will only get 2 marks. And the same applies if you do that with the intraventricular node. All right, so this is obviously transporting animals. We've sort of moved away from transporting plants. And then it says here, figure 5.1 shows features that are observed in transverse sections of 3 types of blood vessels. So, I mean, very easily, you can just think what the 3 types of blood vessels are. You have the capillaries, you have the veins, and you have the arteries. Those are the 3 key types of blood vessels you're expected to know for this level. So there it says blood vessel, wall of 3 layers, wall of 1 layer. This becomes, I mean, quite easy. So if it's just 1 layered wall, you know that means the wall is quite thin, and the one with the thinnest wall is the capillaries, right? And then it says wall of 3 layers, but then it splits that into 2. Thin wall due to lumen diameter, that has to be the veins. The veins have a thinner wall than the arteries because the veins transport blood at lower pressure than the arteries do. And so this is the arteries. And now it says here, I mean, the question is complete figure 5.1 by stating the blood vessel indicated. What this then means is that in order for you to get this 1 mark, you need to get all 3 correctly. If you get 2 correctly but you get 1 wrong, you will not get marks. So make sure you pay attention to that. Then here, the inner walls of the inner layer of the walls of D and E is composed of endothelial tissue. Lists 2 structural features of endothelial tissue. Again, this paper is suggesting that you will have to do a bit more deeper thinking that you have done in past paper 2 papers. So please don't skip anything. D and E, we concluded with the veins and the arteries. And they have endothelial tissue. So what does endothelial tissue do? The endothelial tissue is more or less like a smooth tissue, so it has a smooth surface that allows for blood to travel without any obstruction within these cells, and it is also like a thin layer of cells actually. So it's not bulging or anything, it's just a thin layer of cells that allows for blood to travel with ease. So you can speak about the endothelial tissue. It says list their structural features. They're a thin layer or a single layer, and you can also say they have a smooth surface. Okay. And now we make it to the last question. And this last question is one of my favorite questions. This question has been in many past papers, so I'm so used to seeing it. When I saw it here again, I said, well, at least they've had some kind of mercy on you with this paper. It says in a dividing cell, DNA replication occurs before mitosis. Steps in DNA replication are outlined in this figure, so complete the figure. Helicase allows the enzyme, the DNA double helix rather, to unwind, and the hydrogen bonds between the 2 strands to break, exposing the 4 bases A, T, C, and G. What are the names of these bases? So A is adenine. Please, A is not adenosine. Adenosine is the one that is found in ATP. When you're speaking about DNA replication and all of this, you are speaking of adenine. All right, please don't make that mistake. If you write adenosine instead of adenine, you will not get marks. T is thymine. Okay, C is cytosine, and G is guanine. If we were speaking about RNA, we were speaking about RNA, there would be no thymine, rather there would be uracil. Okay, so just make sure you have that at the back of your mind. An enzyme molecule attaches to each of the two separated parental strands. The two enzyme molecules move in opposite directions, each catalyzing the formation of a new strand of DNA. This enzyme is known as, DNA polymerase. So don't just write polymerase, because if you write polymerase, the person marking doesn't know if you're referring to DNA polymerase or RNA polymerase. So make sure you write DNA polymerase. And if you haven't watched the video I did on what's it called now, on DNA replication and protein synthesis, please go watch that, because that would help you a lot here. I explain the process very easily so that you can easily follow it. Then it says DNA dash, the monomers of DNA are free in the nucleus for the synthesis of new strands. So for the synthesis of new strands, you need DNA nucleotides. Those are the monomers of DNA. Okay, DNA nucleotides. The bases of the DNA monomers form hydrogen bonds with the bases on each separated parental strand of DNA according to the rules of. So when they're speaking about this, they're basically saying A, for example, binds to T, and what causes that? It is complementary base pairing. Okay, so these bases that bind to each other are complementary. So complementary base pairing, that's the rule that guides the binding of DNA monomers, or DNA nucleotides in this case. All right. Oh, and I put the other last part here, I didn't realize. So I'm just going to close this, erase this rather. Then it says one DNA strand is synthesized continuously, and the other is synthesized in sections known as Okazaki fragments. The fragments are joined together by an enzyme called, that is DNA ligase. Again, make sure to put your DNA there, DNA ligase. And ligase is basically the enzyme that is responsible for joining the fragments of DNA together. The result of replication is two DNA molecules, each one containing an original parental strand and a newly synthesized strand. This type of replication is described as semi-conservative, and it's called semi-conservative because you still have half of the original strand in the new strand. So semi-conservative replication. I have no idea what's going on with my pen. I promise you it is drawing things that I have no intentions to draw. But let's move on. Figure 6.2 is a micrograph of a root cell at different stages in the cell cycle. The cell in interface is labeled. So the reason why they've labeled a cell in interface for you is so that you can use that as a reference point to then guide you for all the other parts. Okay, so it then says here, complete figure 6.2 by naming the stages shown in each of the cells, J, K, and L. So if you look at J, it seems as though all the chromosomes are in a single file. So that tells you that this is metaphase. Unfortunately, I can't really explain that in detail. You need to watch the video on chapter 5 if that's a bit unclear. And then K, so if you're looking at K, you need to compare K to the one that's been labeled as the interface cell. In the interface cell, you can see that, yeah, there are these cells here, but you can't really see much. Whereas when you look at K, it seems as though things are becoming a bit more dense and a bit more separated. So that might be the densification of the chromosomes, right, or the chromatids. So in this case, K would be prophase. So that's a good way to think about it, that K would be prophase. And then if you look at L, you can see that the cells seem to be separating, but they don't have separate membranes around them. So in this case, that means that L is telophase, because that's the last phase of mitosis. Okay, so if you want to learn mitosis, the best way to do it is to simply just learn the different stages, like by looking at their images so that you're able to tell them. I would say learn from the images rather than just trying to memorize only the words, because sometimes, or most of the time rather, the questions that come on mitosis involve you looking at an image and being able to decipher from looking at that image that this is the stage of mitosis that this cell is in. Okay, and this is the last set of questions. It says state one feature of the cell in interface that is visible in figure 6.2 that shows the cell is not in early interphase. So they're telling you again to look at this cell, because if you remember from the previous slide, that was our interface cell. So the fact that it's not in early interphase is that, first of all, it has the same size sort of as the cell. If you look at the cell that's in prophase over here, they sort of have the same size, so this is not early interphase. It means that it's already at late interphase, so it has reached the growth point where it is ready to divide, whereas at early interphase it is just about to start growing, so it might not be as large as the other cells. Describe the stage of mitosis shown in cell J. So basically J is metaphase, and if you know about metaphase, in metaphase it's very easy. The chromosomes are aligned at the equator, that's what it's called, so they're in the center of the cell. They're attached to spindle fibers. The chromatids still have the chromatids are still attached to each other, and obviously when you go past metaphase, that is when you have the pulling apart in anaphase. So yeah, that's it from me. I have managed to keep this video under an hour with all the explanations that you need. This paper typically takes you an hour and 15 minutes, so obviously you'll be faster than I have been because you won't have to do all of these explanations. You simply have to think a bit and then write your answers. And obviously this means that you can go back and have a look at what you have done to make sure that your answers are correct. So yeah, that's it from me, and I hope this has been helpful for you guys. I wish you good luck in the upcoming exams. All the best.